Okay, so I guess I'm next. Do I have my 10 minutes, Greg? So, awesome. My group is called the Nanosensors group here at Illinois. And this is an overview of all the things that we do. My background is in electromagnetics and optics. And so in our group, we develop different types of nanostructures that can be fabricated very inexpensively, typically using processes that enable these nanostructures to be made on plastic surfaces so that we can incorporate them in single-use disposable laboratory items such as microscope slides, plastic tubing, microtiter plates, or really any kind of liquid container. Our goal with these nanostructures is to have devices that can efficiently couple with light, such as white light from a light bulb or light from a laser, to concentrate electric fields around a surface. And so I'll show you examples of a couple different projects where we use these surfaces for making fluorescent dyes glow brighter, to get chemical molecules on these structures to scatter light very efficiently so we can detect chemicals at low concentrations. As part of our work, we develop instruments. There's always some kind of machine that we shine light at a structure and then detect light that came off the structure. And some of our instruments are like lab equipment, kind of like this, or things that can be point-of-care detection systems, making like cheap, small, portable detection systems. A lot of our earlier work is with using or making photonic crystal structures, these periodic surface structures made out of dielectric materials that can efficiently couple with and reflect a very narrow band of wavelengths that we illuminate with a broad band of wavelengths, detect a narrow band of wavelengths being reflected. We can build these photonic crystals into biosensor microplates where we can use this structure to detect protein-protein interactions, protein-DNA interactions, where we use this microplate format for high-throughput screening. And so in collaboration with Paul Hergenrother in the chemistry department, we've demonstrated this technology as a way for finding inhibitor chemicals from large libraries of chemicals that can target specific protein-protein interactions or protein-DNA interactions. This technology has been commercialized and is now in use at many large and small pharmaceutical companies throughout the world. We've also done work to incorporate photonic crystals and all these nanostructures into microfluidic channels so we can do these assays with very small volumes. Recently, we've been looking at ways that we can use the internal LED and the internal camera of a smartphone to do a wide variety of optics-based detection assays. And in particular, for this audience, things that can be used in point-of-care diagnostics in agriculture. And so, for example, we've developed label-free biosensor methods for detecting micronutrients in blood samples, so detecting iron and vitamin D deficiencies from a blood sample. We have assays for detecting using fluorescent resonant energy transfer detection of bacterial pathogens. The same technology using a small cradle that has a few optical components in it that couples with an iPhone 4 detection using fluorescence polarization assays for detecting food and grain contaminants such as an aflatoxin detection test. And then same technology can be used for detecting viral antibodies. One type of structure that we make with photonic crystals is a structure that can couple light from a laser to build really big electric fields on the surface of a photonic crystal to make fluorescent dyes that are present there glow more brightly and to channel the emitted light towards our detection system so we can detect it more efficiently. We call this photonic crystal enhanced fluorescence. The basic idea is that we can take fluorescent signals that are weak and then make them very strong. And we've been developing various tricks to make this approach more and more efficient. Where now I think our world record is about an enhancement of around 580 times versus doing the same detection experiment on a regular dumb piece of glass. So we've been making these structures out of plastic, out of quartz, now out of silicon. And so we've been applying this to detecting protein biomarkers in blood samples, doing multiplexed assays for cancer biomarker detection, where we can push the detection limits down below 1 picogram per ml with the enhanced fluorescence. Here just shows the dose response curve for one cancer biomarker, tumor necrosis factor alpha. But we do these in large arrays. We detect lots of biomarkers all at the same time. In collaboration with Lila Vodkin, we've been using the same technology for gene expression detection in DNA microarrays to take genes that are below the detection threshold and to bring them up above it by enhancing the fluorescence signal. And then we've been applying this in our CABPN project to detection of proteins that are expressed by soybeans. So we'll hear more about that tomorrow. And so in our work, we've been developing not only the photonic crystal surface, but also the detection instruments, coming up with fancy ways to make external cavity lasers coupled to the photonic crystal surface, anything to make this electric field on the surface of the photonic crystal to be bigger and bigger. In other work, we've been making nanostructured surfaces such as these arrays of gold domes that we fabricate in arrays that have teeny gaps between adjacent domes. Within these gaps, which are about 10 to 15 nanometers in size, we can couple light from a laser and build an electromagnetic hotspot between these domes, where if a chemical is to enter that hotspot, it would encounter a large field and scatter with a distinct Raman scattering spectrum similar to what Matt was talking about. So we perform this measurement on a surface that we make out of plastic that we can make very inexpensively and detect lots of different things. So, for example, we've used this technique for detecting urea as a urinary metabolite. We've been using the same thing to detect drugs in biochemical tubing. Our CABPN project with Baxter, for example, is to detect pain medications, other drugs, blood thinners that are being introduced to a person through intravenous tubing, where we incorporate the sensor into the internal surface of the tubing as a disposable element. And so doing this, we can detect different drugs. This is a pain medication called promethazine, where we can see the spectrum of promethazine. And we get a response that's dose dependent, and we can see the response going up and down as we introduce and remove or change the concentration of the drug going through there. And so Baxter is interested in this as a method for increasing the safety of drug delivery, to reduce the incidence of drug delivery errors, and for people to know what concentration and combination of drugs are going into a person with an actual measurement. But we can incorporate these sensors really into any kind of plastic container. So if you have interest in detecting contaminants in food, chemical byproducts in manufacturing, all these kind of chemicals have Raman signatures that we can detect. Another project that's involved in CABPN is something that we call photonic crystal enhanced microscopy, where we have a photonic crystal surface that we make on a microscope slide, where this microscope slide is special from the standpoint that we can use it to detect cell attachment on the surface. So in any place where a cell attaches will reflect a slightly different wavelength than the places where the cell is not attached. And so once we can attach or detect a cell attachment, we can now use this as a platform for detecting all sorts of things that you can do to cells. And so for example, we can detect cancer cells reaction to drugs. We can monitor and image the surface attachment during stem cell differentiation, cells crawling and moving across the surface, such as involved in wound healing, and looking at various drug-cell interactions. And so this is just an example of a highly detailed image of a few cancer cells on our surface, where we can see the change in wavelength that occurs when the cell responds to a drug. And so in terms, one application in that's of relevance to CABPN is we've used this as a platform for detecting a whole library of plant extracts from a bunch of exotic plants in Pakistan and India. These are extracts taken that are purified, sent to us. We grow pancreatic cancer cells on our photonic crystal, and then image their response as they undergo apoptosis as they're exposed to different plant extracts. We find some extracts that actually kill the cells, other ones that don't do anything, and then some that even make the cells grow faster. And so we use this as a screening method. And so I think I've used up like 9 of my 10 minutes. So I'll be happy to answer questions, or if you have interest in any of these topics, we'd be happy to discuss them tonight at the open house. Okay, so let's move on to